Okay, so we talk about microbial growth. We finished talk about here. Then we're gonna go to the lecture 5 slides. Lecture 6 slides are gonna using another resource. Okay, I'll put there. So we talk about the culture media. Under the culture media, they could be classified with chemical constituents, physical nature, and function. So this is a very good slide for the review. Based on the chemical composition, we have a defined media. We will call it a synthetic media, complex media. The physical nature, we have a liquid, semi-solid, and solid, and support media, enrichment media, selective media, differentiate. We call it based on the function. And this is give you some of the example, and I have a question for you. If you have a glucose, peptone, tryptone, even give you amount, is that a complex media or defined media? The answer it should be, it is a complex media, because we don't know exactly inside the tryptone and the peptone what type of the chemical it is. But what are the examples of a defined media and a synthetic media, which is right on the top, and you can see all those chemical formula there. So that's why it is a little bit tricky there, okay? So defined, which means all the chemicals are clearly defined. Then we give you example, what is peptone, what is extract, what is the agar. And then we mentioned about the support media, support bacterial growth. Enrichment media, you add some of the ingredients, let's say sheep blood, and make it more nutritionized, and then can grow bacteria dramatically. And then also the nutrition blood agar, we can heat it at 70 degrees Celsius and turn it brownish, we call it chocolate agar. And we also give examples of the selective media and differentiated media. So a very good example is a McConkey agar. And we say McConkey agar, first of all, it is a complex media. It's a solid media. Then we talk about a functional. It is a selective media. Why? We have crystal violet there. And crystal violet is a selective agent, because only gram-positive bacteria, only gram-negative bacteria will grow, gram-positive bacteria will be killed. And we know the reason, because crystal violet will attacking peptidoglycan. The bile salts is also a selective agent. This is a picture for the McConkey agar. Okay, we can see the lactose use, which is turned this brownish color, pink color. And we also say, because there is a lactose there, so McConkey agar is a differentiated media. We could differentiate with lactose fermentation positive and negative. And then we talk about the McConkey agar, lactose fermentation, non-fermentation. Then we talk about the blood agar. It's a very important slides. We will mention later on when we go exam 2. So we have an alpha hemolytic streptococcus pneumoniae, which is dark green color. It is incomplete hydrolysis, hemolyzing of blood cell. And you have a beta hemolytic streptococcus pyogenes, which is called strep throat. And you see the transparent zone, which means completely hydrolyzed of the blood cell. So this tells us blood agar is a differentiated media, because it can differentiate hemolysis. But obviously, it is an enrichment media, because it has a blood there. It's a higher nutrition ingredients. Okay, now let's talk about several other technology. The first one we want to talk about is isolation. We can say isolation and numeration. Okay, isolation, what we do is we call streak plating. So what is a streak plating? We mentioned about that. We want to get a single colony. So what we do? We're using a loop to touch the bacteria. Let's say there's a bacteria. We're using a loop, go inside, take it out, And then on the edge of the first college, we do a very intensive streak. Then we have to flaming the loop to killing the bacteria. From the edge of the first college, we do a less intensive one. And then from the edge of the second one, we flame in the loop, we do a third one, third college. And the last section, we flame the loop again and do the last one. And then we incubate 35 degrees Celsius, 24 hours. What are you gonna see? We hope to see the colony there. Those dots there. And that is a single colony, which is a group of bacteria coming from one mother cell. We call this is a pure culture. So what this tells us, the streak plating. Streak plating will tell us in this bottle of water, is there a bacteria or no bacteria? So streak plating's purpose is we can test presence, absence of bacteria. But we don't know the exact number. So we call this is a qualitative test. Qualitative test. Okay, how do we do the exact number? That's called numeration. There is two methods we can do is pour plating and spread plating. Now that's next to what we do. Now, by the way, that's a streak plating. Okay, you see the streak. Every time we coming from the edge of the previous streak, and we do 4 quarters, and then we do the incubation. You see the colony will be forming, and this tests the presence or absence. Okay, spread plating and the pour plating. How we do? What's the difference between that? Let's do spread plating first. Spread plating, which means you have agar plates already. And I'm going to add a liquid there. The liquid usually we add 100 microliter equals 0.1 ml. Then we using a plastic sterile, which means already pre-treated, no bacteria there. Spreader to do the spreading. So let's say you're going to do like this. Okay? That's called a spread. Averagely spread on the surface. And these agar plates, we grow 35 degrees Celsius, 24, 48 hours. It depends different bacteria. And you will see those colonies there. And then we count. And this is what it looks like. Then we manually count those colonies. Or you can put in a machine. The machine can scan in how many colonies it is. And sometimes we compare the manually count and the machine counting, we see there's a difference. The scanning machine usually is expensive, like $5,000 sometimes. Right now, a little bit decreased. The new one could be $5,000. So we count everything. It's a single colony. So we manually count it. Now there's a remaining question. Okay, I have a bacterial broth, let's say like this. Okay, this is E. coli broth, let's say. Very turbidity. Okay, I'm going to add there 100 microliter. Then I do the spread. Do you think it works? It works? Probably not. Because it's too heavy population. You're not going to get those single colony. Not going to be like that. So before you do, we need to do 10-fold or 100-fold serial dilution. And this is, some of you really doesn't like it because it's in the chemistry class. It's always like a tricky. You do that. 10-fold, we know 1.0 ml go to 9.0 ml, say multiple times. Is that right? We do these tubes a couple of times. 100-fold, 0.1 ml into 9.9 ml, couple of times. Now, what's the solution we're using? 0.1% buffered peptone water. It's a dilution. Because we want the bacteria is not has a loose osmotic balance. So we don't use distilled water, we're using this buffered peptone water. Okay? Therefore we get those single colony there. That's important. Now every single colony when you count it, we call it CFU, stand for colony forming unit. Now here is a question. Sometimes in my other class in the lab, I have a plate comes out. I said you go ahead to count it. And lots of students don't want to do it. We just sit there. Because it's too many. Okay, that's reasonable. It makes sense. So in the food microbiology or general microbiology area, we have an acceptable zone. Which means on these plates, if you have 30 to 300 colony forming units, these plates, we will use it. We say it's acceptable. How about if you only have 10 colony there? We don't use that because that 10 colony could come in from cross-contamination. If we have a plate have a 500 CFUs there, are we going to use it? We of course not use it. We call it TNTC, equal too numerable to count. So what we have to use the next level of the dilution because theoretically the next dilution will get 50 colony. And the 50 is well within the 30 to 300 range. So that's why the dilution technology is very important. Okay, that's spread plating. What is the pour plating? Spread plating is we have agar plates already made. We made it agar. Okay, let's say like that. What is pour plating? Pour plating is we had an empty petri dish. That is empty petri dish. And I'm adding liquid. Now what are we missing? The agar. At the same time, I'm adding 20 ml melted agar. Then we mix it. Wait for solidify. Because agar, melted agar, at 25 to 30 degrees Celsius will solidify. That's why I tell you one thing, Fanny Hesse, she's really smart. And this material is a magic when you've find it like 300 or 400 years ago. Otherwise, you're not going to get any bacteria. Because in the solution, you can only see the turbidity. You're not going to get a single colony. That's why it is a magic. Okay, when you mix it and then wait for solidify, then of course you have to do incubation, and then you count the colony. But so what's the problem for that? What means melted agar? Melted agar, which means you need a little bit of heat there. You can use like 30 degrees Celsius, it's going to solidify immediately. So melted agar, if we are using melted agar, which means this agar has a mild heat. The mild heat, 50 degrees Celsius, sometimes 55 degrees Celsius, and they're going to kill bacteria. So, pore plating, basically you get a lower number compared to spread plating because the mild heat is killing the bacteria. So that's not very nice. But what the good thing for pore plating? Spread plating, we need 1 day to prepare the agar. We pour it, we wait them to solidify. That is 1 day time and the labor. At the pore plating, you can dumping together with liquid melted media and put the bacteria there. That saves you 1 day energy and the labor cost. And that's for industry is important because it's all the time and the money. So that's why they do the pore plating all the time. So pore plating basically they give you an estimation of the number, how bad it is. Okay, so for example, if I have a food products which is contaminated with Pseudomonas, a sporage bacteria. I don't use an example of a pathogen because lots of pathogens are zero tolerance. Then I need to see how bad it is, so I spread the plating. But I think I lose the labor. Tomorrow somebody is not going to come in. What I do? I do pore plating. After pore plating, I get to see if the pore plating, I find the colony is TNTC. Obviously, that's really bad, the sporage. So we need to reevaluate our sanitation standard operation procedure. Is there something wrong with it? And sometimes when you see the sporage bacteria grow on the plates, you also will see those packaging has a bubble comes out. The gas generates. Because during the sporage, there's a little bit of fermentation happens, and the gas is created, and also the acid is created. So that's interesting why they're using the pore plating. To save the time, give you a briefly estimation. And this one is, they explain to you what is the spread plating. And you can see the picture there, and how we do the dilution. So, I'd like to let you try. So, at the early May, and in our second last class, we will do the spread plating, pore plating, and streak plating. Then you're going to get the colony. Is that right? Then what are we going to do the colony? In the last class, we're going to go to the lab. When the colony, we could do gram stain. Then you clearly know what the bacteria it is, what it looks like. I will do the half of you guys do Staphylococcus aureus, half of you do E. coli. Then you see the difference between gram-positive and the gram-negative bacteria. So that's the last 2 classes what we're going to do. And it's a very basic technology using widely in the lab, in the microbiology lab. Okay, spiral plating. This is a tricky one. First of all, this machine is expensive, $20,000. Okay, how they do it? You look at here. This is we call it a vacuum pump. When you press the button, it gives you negative charge. It gives you negative vacuum, negative pressure, because these pipes gonna be absorbed. And when you see here, the first one is usually a liquid. The first one is usually 70% alcohol. They do the sanitizing of the cleaning of the pipes. Then the second one is usually a water to rinse. The last one is a sample. And when you get the sample, you press the button, it's to the steel, stir. This is a picture what they're using is crystal violet. You can see it very clearly, the circle there. And then gonna do the incubation, and after incubation, you see the colony like that. And then you go to the scanning machine, will tell you. So, why people use this? Well, it certainly saves you labor. because you don't have to do, one person can handle this. The major reason is that using pore plating and spread plating, we need lots of dilution. Pore plating and spread plating, at least 3 or 4 tubes. Spiral plating, you only need 1 sometimes, because it's a dilution covered much more. It's covered 10 to the power of 1 to 10 to the power of 4. If you do not do any dilution, which means it saves the dilution sketch, which means it saves the procedure. However, there is a limitation. Spiral plating, you can only testing liquid products. You cannot test food because the food particle will stuck there. And basically it's a filtered water solution. Even like juice, you cannot have a pulp there. If you have a pulp there, it's going to stuck the tube. It's very thin one. Okay, but people still do the spiral plating because they cover multiple dilution factors. Make the procedure a little bit easy. But I also want to tell you that stirring, it took 1 minute. So very slow. You put 1 plate there, it's going to stir. Then you put it out, that took 1 minute. And also depends on the volume. If you are not using 100 microliter, you are using 500 microliter. Finish 1 plate stir could be taking 2 minutes. So that's very time consuming and slow very slow procedure. Okay, but they have a commercial product, $20,000 for spiral plating. This is the old time. If we don't have that scanning machine, you have to compare the color table. That makes it very tricky to count. Okay? Okay, this is important. We talk about the gram staining. We talk about the gram positive, gram negative. We talk about the large cork cell. What this means? Those are we call cell morphology. What means morphology? Shape of the cell. On the other plates, you also gonna see the morphology. That is called colony morphology. And that is also important. So what are they? You can see the form. That could be punctiform, circular, filamentous, irregular, rosal, and spiral. Elevation, it could be flat, raised, convex. The margin could be entire, undulate, locubate. So colony form morphology also tells you some of the information for the bacteria. A very typical example is a pushable colony for Neisseria gonorrhea. So Neisseria gonorrhea grow on chocolate agar. It's a fastidious bacteria. When you look at the colony morphology, it's a pushable colony. It's Neisseria, very possible. So that's why I tell you sometimes the colony morphology is also important. Even sometimes we can smell it. The colony maybe have some smell, have some different smelling stuff there, the chemicals there. So that's something else. Okay, how we measure microorganisms counts? There are lots of methods. We mentioned one is only agar plates. This is called direct colony counts. We could do is using a chamber. So we have these called, this name we have Petrohoff-Hossy counting chamber. It's very similar to count your blood cell. You put in the chamber, the liquid, cover with glass, and underneath microscope, you are counting. It's very good because it's easy, inexpensive, and quick. It can count in both prokaryotes and eukaryotes, which means bacteria and fungi both can be applied. However, what is the disadvantage? You cannot differentiate live cell and dead cell, because everything is there. Spread plating, the colony here is viable. Those are live cell. Dead cell will not show up. By using this, you will not know live or dead cell. Okay, another method, I believe lots of you did. Using color spectrometer or spectrometer just. So a bacteria growing here in the tube, and we are talking about 3 indications for bacterial growth. What are they? If you have a bottle of water, let's say this is juice, you have bacteria on the top. We call it pellicles. That means something's growing. You see the whole bottle is turbidity. That means something is growing. And also you see the precipitation at the bottom. That means the bacteria is there. And if you shake it, it turns turbidity. Or using layman language, it's cloudy, which means bacteria is there. So how we do, we put in a color spectrometer and we adjust the wavelengths, we give you absorbance value. Obviously, the higher value, the more bacteria theoretically present. However, what's the disadvantage of this method? Again, you cannot differentiate live and dead cell. So let's say this turbidity tube. It could be all live cells, it could be all dead cells. But why this is important? Because it gives you an idea how many it is if you assume those are live cells. If you see very high turbidity, it could be around 10 to the power of 8 to 9 cells per ml. So this method, we call it a semi-quantitative test, because we cannot differentiate live and dead cell. Okay, how about we do modes? This is a filtration method. So we have a membrane filtration on the water. And we put the water there, then they go to the membrane filter. The membrane filter is 0.45 micrometer. The eastern modes could passing through. Bacteria will pass it, cannot. Bacteria will prevent it at 0.22 micrometer. So using this, eastern modes could passing through. And then you put a membrane system into your agar plates, and in incubation, you will see the colony is forming. This is a very typical for the system called ISO green. I did this test 15 years ago. My first job working in the company, we testing the water products to see the water, municipal water, drinking water. We want to see whether there's a bacteria there. Because in here, the EPA, Environmental Protection Agency, the regulation for drinking water in the United States is zero tolerance for coliform, fecal coliform, and the microorganism basically. So we using this method to do those disease sample. And you going through the funnel, the funnel is negatively charged, it's very loud voice about that machine. And then you put a filter paper, you going through the liquid, then you put the filter paper, use faucets, sterilize the faucets, put on the agar plates, and then you can see those colonies. It is very good. Why? You know the eastern modes, it could be very crazy. Or grow on the agar plates, you cannot count it. The colony looks like fuzzy. But using this filter paper, regardless of what type of the eastern modes, they will form the colony exactly the same as bacteria. So it's very easy to count, to do the numeration. That's why the ISO grid is used widely in the industry to do the numeration of the eastern modes. And another one important is Legionella. That's another human pathogen for Legionella, and that has to be zero tolerance too. And And then using this method also we can test it. Okay? So that's very interesting. We talk about all these. So we finished talking about here. I haven't uploaded the slides here. I need to go back to the last year. I'm gonna using the slides from last year. We're gonna see. So this is last year's class. We had slides number 5. We want to talk about that. So we're gonna use. I'll upload to the when I go back to the home. So okay, that's bacterial median. It should be number 6, I believe. Let me see. Okay. Seems like right here we are. Okay, bacterial fidelity. Did we talk about the control? Okay. So we finished the talk about here. Let me go back. Okay, all the material we actually finished because I double check with last year's slides. So what's the plan? We have an exam, is that right? So exam will be open book, and the open book will be you can do by yourself anytime and during the classroom, the class time, and you cannot talk to other students, but you feel free to do all the using all the resource. Very important thing I want to say is that no texting. Okay, you cannot text to the other people. So what we're gonna do? We're gonna do a review. Exam will be on next week Tuesday. This Thursday, my PhD student Corey will give a special talk about feeds. Chicken feeds, microbial safety. He did a lots of research about thermal inactivation. It's a lot of fun, so we can talk by the time, okay? So we get about half an hour. Let's just go back to do the review, what we have. Because I believe it is important. And something possible in the exam, I'll let you know. So preparing open book exam, what you need to prepare. You don't have to remember. That gives you lots of the less burden. Because otherwise you have to remember. You don't have to. But you need to know where it is. And that means it tests you the skill to find the information if you need it. That's why I think it's worth to do it. Okay, what is microbiology? This is easy. If I have a question, what is microbiology? You just go here. There's microorganism, algae, bacteria, virus, protozoan, fungi. This is important. Is that right? If you ask you, what is the varioids? Right here, composed by RNA. What is prions? Composed by protein right there. Okay, what is protists? Protists, sorry. Algae, protozoan, slime molds. This slide is important to give you the example. What is this? Tells you some bacteria really bad. This is one be example, good example. Start from here. We maybe ask you to corresponding to the person, their achievements in history. So Edward Jenner, what is the keywords here? Vaccination, because protecting individuals from smallpox. What is vaccination? That's vaccination, okay? What's the purpose? Active immune. Examples of the vaccines, right here. Who is the first person discovered the microorganism and recorded? Is Anthony van Leeuwenhoek. So that's a very simple question. Who created swine neck? Louis Pasteur. And he, from microbiology standpoint, is repel spontaneous generation. That's a good example. Okay, John Tidal and Ferdinand Cohen, they both clean the mess, you can say, because there is a description about that. heat resistant, and endospore forming bacteria will make the results look different. Bob Koch. What is major contribution? Koch postulate. So if I ask you what is Koch postulate? 1, 2, 3, 4. You don't have to exactly copy the original words. You can paraphrase it. Okay, as long as you have a key points, we'll give you 4 points. You don't have to silly like, from beginning to the end. You may be spending 10 minutes to just writing the words. So you can use your own words to describe it. Pure culture, that's important. So Fanny Hesse is first one suggests use agar as a solidifying agent. What is agar? Polysaccharides from seaweed. Okay, streak plating we just mentioned. Very important, naming classify microbes binomial nomenclature, species name, genus name. What is the exception? Is Salmonella, because Salmonella we'll talk about subspecies. It's not a species name, it's a subspecies name. And we may not test you that difficult. That's usually a PhD defense question, but we just want to let you know the knowledge about that. Okay, that's lecture 1. It's basically easy. Then we go talk about lecture 2. We just go over it, because all the time when we go through the exam, we want to review. We want to know where it is. Okay. Light field microscope. Examples, electron microscope. This just mass, we won't be testing you too much. This tells you light is refracted. Lens, this is important. Short focal length equals more magnification. Okay? This tells you the working distance. Decrease with increasing magnification of the lens. Shorter wavelengths, greater resolution. Basic structure of the bright field microscope right there. We want just to ask you that. How to calculate total magnification is important. It's a product of ocular lens multiplied by objective lens. Okay, and you also need to know, I may forgot to mention that bright field microscope has a very good characteristics we call it a parfocal, which means once it's focused, it stays focused. You only need a very minimal adjustment. Oil immersion, remove the air gap. Dark field microscope, there's a block, so light going through perpendicular. The difference between bright field and dark field. Why it is important? Treponema palladium is a pathogen for syphilis. It's a spirochetes, special bacteria. Phase contrast microscope, because of the phase rate, so change the wavelengths. Little bit of physics behind that. We won't test you that deep. Why is this important? We can test in the internal structure of the eukaryotes. This is one I think is very clear. You can see the difference between light field, bright field, the phase contrast, and the dark field microscope. Phase contrast, you can see the internal structure much more clear in here. Fluorescence microscope. What's a major difference compared to the light? Energy resource. It's coming from ultraviolet, violet, and blue light. Okay? You need a stain in DNA, so we're using daily stain because we can differentiate live and dead cell. That is important. Confocal microscope. Why it is important? We know 3D images. Why 3D images is important? Because we need to testing a special bacterial matrix called biofilm, because they are resistant to the sanitizer. You don't have to worry about this. This is an example. Electron microscope, we have 2, transmission scanning. Transmission, internal structure, scanning is on the surface. 3D images. Okay, this is different size of the bacteria we mentioned. Difference between light field, electronic field, the difference. That's a good comparison. Okay, if you ask a question, that's a good slide to be seen. Transmission electron microscope. specimen preparation, negative staining, shadowing, we mentioned about that. Freeze etching, we mentioned about briefly, 3D images for the scanning electron microscope. You don't have to worry about these too much. This is the experiments. Okay, so lecture 2. Lecture 3, what we talk about? If you remember, very important is staining technology. So we prepare a smear, air dry heat fixing, add a dye and do the exam. Okay, very, very important. The air dry has to finish it about 3 to 5 minutes. Otherwise you have a lag there. Heat fixing is to killing the enzyme, let the bacteria stick on the slide surface. And that's the first procedure. Then we talk about the staining technology. We have a simple stain, we have a negative stain. Simple stain, we're using the dye, crystal violet, saffronin, methylene blue, carbon fusion. Negative stain, we have a nigrosin, Indian ink, rose bengal, and acid fusion, all these things. Now, simple stain, we are staining the bacteria. Negative stain, we are staining background. And this is a very easy one to tell you the simple stain. You can see the bacterial morphology and arrangement, which means the size, shape. This is a good example for negative stain. This is a capsule. Okay, you see the capsule. And you see the diprococcus cell. It is Streptococcus pneumoniae, but it's a good example for negative stain. And the background is completely dark. Then we jumping into a very important staining technology. Askew gram stain procedure. Very simple. Just find the slides. Crystal violet, followed by grimidine. What's the function of grimidine? Is a mordant to let the crystal violet staining strong. Then alcohol. What's the function of the alcohol? Decoloration. That 10 seconds is very important. Counter stain with saffron. Okay, there is a 2 easy question for you. When the gram stain, we're always staining like a gram negative. We call it a gram variable stain. Number 1, we're using an older culture. Number 2, is decoloration too long? Is that right? If your decoloration is 3 minutes, everything is gone. Think about if you have a dye on your hands, what you do, using alcohol. So that's gram stain. Okay, why gram stain stains purple for positive, pink for negative? Well, that's related to the cell wall structure. Gram positive, you have a very heavy peptidoglycan. Can hold on the dye, so did not lose during decoloration. Gram negative, peptidoglycan is very thin. Plus, there is LPS, lipopolysaccharides. The major components of lipopolysaccharides is lipids A. That's important because makes a cell system a huge amount of the lipid, and the lipid dissolve in the alcohol solution and is easy to be washed off during decoloration process. Okay, so these are important information. Acid fast stain. Which bacteria is for acid fast stain? Mycobacteria tuberculosis. Why we have to do acid fast stain? Because it's high lipid containing the cell wall, with the reason is higher components of myconic gases. Very waxy cell wall. What's special for acid fast stain? Heat steaming. Because we want the dye can penetrate. Plus we have to use acid alcohol. Okay? Endospore stain, we talk about here. What's the function? Is there endospore? Where is endospore? Because the location genetic dependent. By the way, endospore is not, we call it a reproductive function. It's a protective function, which means the mother cell has endospore does not mean the daughter cell automatically have endospore. Okay, capsule stain, you can do negative stain, or you can using a carbon sulfide. Flagella stain. Right here, I can see the flagella because flagella related to the true bacteria or microorganism motility. This is a very good example. Okay, we use the gram stain, acid-fast stain, endospore stain, capsule stain, and the flagella stain. The bacteria has a major flagella covered on the surface. It's called Proteus vulgaris. Okay. Why we need to do the staining technology? We need to know the shape and arrangement. So what are the shape? Could be coccale, diprococcale, streptococcus, staphylococcus, tetraid, sarcini. What's the shape of others? Bacillus. Lord's shape, vibrio, spiral sheets. Okay, so that's the second one. Not difficult, it's basic knowledge. Exam 4, or exam 1, lecture 4, we have a very important slides right here. Are very special for bacteria. It's binary fission. Therefore, have a concept for generation time. Is time required for a population to double in the number? So we have E. coli double every 18-20 minutes, very short. Mycobacterial tuberculosis double very slow. That's why we have to use acid-fast stain. Now, by the way, if acid-fast positive, does that mean this patient has TB? No, it's presumptive TB, but we have to be using antibiotics because the generation time is so long. If we get a pure culture, it took forever. Absolutely in the exam, bacterial growth curve. We'll ask you to draw it. Is that easy? Open book is always easy. You don't have to remember. So this slide is important. What's the key? Some of you forget to label x-axis and the y-axis. Make sure you do that, okay? We need to know which one is which. So x-axis time, y-axis population. Then you have a lag phase, preparation, log phase grow dramatically, the stationary phase, growing death is the balance because waste and nutrients is the balance. Then later on more toxication there, so death phase. It's very special for microorganisms. Factors affect microorganism grow. pH, temperature, water activity, oxygen. Okay, what is the pH? That's easy.